Hi everyone, welcome back to the ChIP-seq data analysis. In the last lecture, I've introduced the ChIP-seq technology and how to use MAX to detect the DNA binding sites for the DNA binding proteins. In this lecture, I will introduce how to use another peak calling software called Sighsir and also how to map the peaks to the genome. MAX is widely used for narrow peaks detections. As I mentioned before, the histomodification peaks are larger and lower. Therefore, the peak caller should be able to detect the large peaks consist of widespread tags. Sighsir was designed to handle such tasks. It's written in Python and can be used on all three platforms. In addition, the Python package NumPy and SciPy is required to install for Sighsir. I borrowed a figure from the Sighsir paper to show the essential idea of the algorithm. Sighsir will partition the genome into windows and then count the reads in each window. If the total reads pass a threshold based on the statistical model, the window will be marked as an eligible window. Next, Sighsir searches for regions that are enriched for eligible windows, while also allows several ineligible windows in between, defined as gap. In this way, Sighsir is able to identify large island domains consist of broad permissive read signals. The size of the window and gap can be customized by the user. This is the command line to invoke Sighsir. Unlike MAX, the parameters for Sighsir are at fixed positions. They are passed to the Python script through a master shell script. Therefore, in the Windows system, we need to use Sigwin to perform Sighsir runs. Use sh to execute the Sighsir.sh shell script. You can also modify the path to Sighsir in the shell script. The treatment and control dataset parameters are self-explaining. For species, we can use terms such as HG19 and MM9 to indicate which genome we use to map the sequences. The redundancy threshold is similar to the keep duplicate parameters in MAX. Here we use 1 as recommended, and Sighsir will throw away all the other duplicate reads. The window size specifies the size of the scanning window. It's usually set up together with the gap size parameter. The authors of Sighsir suggest to use 1-to-1 ratio of window size to gap size for narrow peaks such as H3K4 trimethylation and 1-to-3 for broader peaks such as H3K27 trimethylation or K36 trimethylation. The fragment size points to the size of the tags. Since we know the size of the nucleosome is approximately 150 BP, we put 150 here. The effective genome fraction is similar as the G parameter in MAX. The authors of the Sighsir have provided this number of a human and a mouse. For other species, we need to estimate this ratio based on the reference methods that Sighsir provides. The last parameter is the FDR threshold to call significant peaks. Sighsir uses 0.05 as default. You can change it to a more strict threshold such as 0.01 or 0.001. This is a screenshot as Sighsir is running. It also prints the parameters at the beginning. Sighsir will then remove the redundant reads and show the stats for each chromosome. It then partitions the genome in windows according to the window size we give and look for candidate islands/peaks. Next, Sighsir calculates the significance of these candidate islands using the control library and keep the ones that pass the FDR threshold we specified. The last few steps is to obtain the reads that consist the significant islands and calculate the average intensity for each of the islands. Sighsir will generate alpha files in BAT and WAKE format for the convenience of the downstream analysis. At each step, Sighsir will show which Python script is being used. So if the program breaks, we can go back to that specific script and check what causes the problem. Also, I highly recommend to save the on-screen print to a log file for both MAX and Sighsir. It will remind you about the parameters if you need to repeat analysis or just to check for the parameters in the future. These are the output files from Sighsir. The Sighsir user manual provides details about these files. Here I want to focus on 3 of them, which are the most useful ones for downstream analysis. The score island file contains the significant islands when running Sighsir without a control dataset. If you do include a control dataset, the corresponding output file you should look at is the island summary FDR file. It gives a complete list of all candidate islands with their statistical significance. The island bed file contains the chromosome coordinates of only the significant islands and the rate count for each island. Now let's open the island summary file. For each candidate island, we have the chromosome coordinates, the start and the end position, the number of reads mapped to the island in the treatments dataset, and also the number of reads with the same location from the control dataset. The next columns hold the p-value, the fold enrichment, and FDR for this island. These features are common among ChIP-seq peak calling software. We can tailor the threshold for the p-value, the fold enrichment, and FDR to narrow down the number of islands for further analysis. Once we get significant peaks or islands, the next step is to find the nearby genes. Many softwares are available to annotate the peaks, and they are relatively easy to use if you are somewhat familiar with the common tools such as R, Python, and MATLAB. For example, an R package called ChIP-seq Anno is able to associate the peaks to nearby genes and also categorize peaks according to their nearby genomic features such as transcription starting site, upstream of TSS, gene body, etc. In this and the next slides, I provide a general framework of how to do it yourself, mainly because the pre-made software limits the flexibility. You will have better manipulation of the dataset if you can play around with your own code. To associate the peaks to nearby genes, we first need to download the gene annotation files from the genome browser. The most recent genome build for human is HG19 and M10 for mouse. On the FTP client, the redflag.txt file is what we need. It's a tab-delimited file, and here I'm pasting the documents explaining each field. For each gene, it contains all known transcript of that gene and the transcription starting and end position, the code region start and end position, and also the start and end position of each of the exons. To bear in mind, the strand on which the gene gets transcribed matters when we map the peaks to the genes. The order of the start and end position is reversed on the minus strand. And also, for genes that have different isoforms, you need to make decisions about how to handle these isoforms. Now we have genomic annotation of the genes. It's time to choose your favorite programming language and brush up your coding skills. The workflow is the one I've used. I put here as an example for your reference. I encourage you to develop your own workflow according to the specific biological questions you try to answer with the ChIP-seq experiments. Since I'm not interested in the difference between isoforms, I use the longest transcript to represent the gene. The idea is to use the locations of genes as the anchor region and identify peaks that overlap with this anchor region. Usually, we will allow certain distance from the anchor region to peaks, such as 2K, 5K, or 10K. Therefore, I extend the anchor region's coordinates according to the distance, and then construct a hash map type of variable to temporarily hold the positions of all anchor regions. In the meantime, the script will read in the peak positions from the output of max or Sightsir and pass to a hash map or dictionary variable. The next step is to loop over each peak to each anchor region, and if they are overlapped, write the peak and the gene to the output file. There are many details to think about when you're writing the code, for instance, how to define overlapping, and there is probably no uniform answers. Here is the example program I've wrote. You can download from the course webpage. Uncompress the package and we'll see pre-organized directories. I'll put the genome annotation files in the genomes folder for your convenience. The next step is to run the pick-to-gene draw program in the command line. Both Windows command prompt and Mac terminal work. The details about parameters can be found in the README file. The output files can be found in the output folder. You can also define your own output path in the command lines as an argument. The GeneMap and PeakMap files contain the same information but in different format. For example, the GeneMap file lists all peaks that map to the same gene in order of genes. The PeakMap file lists genes that overlap the peaks as well as the distance from the center of the peak to the transcription starting site to the nearby genes. The distance can be used to rank the peaks. I've shown how to use Max and Sightsir. Except for these two, many other softwares are available. This table lists some of the popular ones and the functionality of each. Basically, it's a good practice to use a peak caller with window-based scan, statistical model or t-test, and gives a false discovery rate. Based on their algorithms, even given the same dataset, each software will identify similar but different peaks. Trying different reliable peak callers and take the consensus peak is also a good strategy if your time and resources allows.